Okay, I guess I don't need anybody to introduce me anymore because you're all getting very tired of seeing me up here. My last lecture, very nice. And today I'm going to sort of finish the general area that I talked yesterday. Yesterday was about what I think of as physical effects of the DNA on transcription, and by affecting transcription, of course, it affects network behavior, and the particular aspect of network behavior, which seems like is connected to those physical properties of DNA that become important during transcription, have to do with the transcriptional noise. So that was what I talked about yesterday. Today I'm going to give you, because it's been at least a day since you've heard about EMT, I'm just going to review very, very quickly some of the ideas about EMT, and then proceed to talk about the part that's missing in all those ideas, which has to do with the epigenetic layer of regulation. So let me just very quickly review some of the things you heard about in my general talk, in Mohit's talk, and other people's talks, which has to do with the fact that starting from around 15 years ago by now, the cancer community adopted the idea of EMT. They adopted it from the developmental biology world, where it was well known that cells could undergo transitions from epithelial-like behavior to mesenchymal-like behavior. And the cell which has been most studied in that context is the neural crest cell, which is responsible for leaving the neural tube, the part of which will eventually develop into the spinal cord and the brain, and going off to the other parts of the body and differentiating into a bunch of different types of cells. So the cancer community adopted this idea to be able to explain how it was that cells would leave the primary tumor and progress throughout the body, reach the circulation, and then eventually land somewhere else and begin to grow secondary tumors, i.e. metastases. So here is just a cartoon picture, and the idea about EMT is that it sounds like it's about motility, and of course that's how it was originally discovered and described from those periods of time, but it's really a very large-scale change in the complete nature of the cell. So here is just an example, a very early example from, again, 15 years ago, about what happens when a cell undergoes EMT, when you just say, oh, it's, what changes? And the answer is, well, sort of everything changes. So here is, for example, a list of proteins that change. Some of them are obvious, like ECAT-herin is the thing which sticks epithelial cells together, and that gets downregulated because mesenchymal cells have much less adhesion than epithelial cells. But there are all sorts of other things here, some of which we understand in detail what they're doing, some of which we don't. And then on this side of the ledger is all these other things which become expressed in the mesenchymal phenotype. And there are things that that are integrins, which allow the cells to effectively attach to matrix materials and propagate and move through the body. There are, of course, all sorts of other things. There's changes in the actin. So what you're supposed to think about when people say the word EMT is it's not like there's a couple of genes that change and now you had two markers that were epithelial-like and two markers that were mesenchymal-like, and those things changed and that somehow allowed the cells to move. It's actually a complete change in the shape, which we saw yesterday, in the mechanics, in the spatial organization. Many, many aspects of the cells change when they undergo EMT. So it's important to realize that when someone asks the question of, is EMT necessary or important for metastasis, it can be important for metastasis along a number of different directions. It can be important because it enables motility. That's what was in that cartoon. But it could be also important because for some reason, cells undergoing EMT might be more resistant to immune systems. For example, some number of years ago, we showed that cells that underwent EMT downregulated some of the machinery that's involved in displaying peptides on your surface. It downregulates the ability of the immune system to recognize foreign cells. So it's not listed in this property. It was only discovered by us together with a lab at MD Anderson at some point. But it shows that there are many aspects, and it's not completely clear when you say EMT is important or EMT means this. It's not clear you've captured the right aspect for what you're thinking about. So that's always to keep in mind. Now, when we study the system, and I said I'm only going to go through this. We have all these networks. This is a a network of created by Rika Albert, and it has various pieces. I mean, the way you think about these networks is there are inputs coming from the other cells. So if you look at the top lines here, there's TGF-beta. Many of the laboratory experiments on EMT use TGF-beta as a pharmacological inducer of EMT, but it's not the only choice. You know, you can almost find all your favorite pathways. You're interested in EGF, which we heard a lot about. If you're interested in the Notch pathway, if you're interested in the hedgehog pathway, all of them can be used under the right context to drive EMT. So just like there's a huge range of different things that change, there's also a huge range of different things that can induce EMT, which depending on the context that you're talking about. And then you have this network which couples all these various things together, and eventually down at the bottom here, you have something which makes the choice between becoming epithelial, becoming mesenchymal, shifting from one to the other, depending on all those inputs to the cell. Now, when you want to study this model from a mechanistic point of view, it's very hard to start with something even at this level of complexity. Now, when I put up this diagram, I always like to say that this diagram serves as a very simple test that you can give to somebody and see if they're coming from a physics background or coming from a biology background. If they're coming from a physics background, they look at this picture and say, how can you ever hope to understand something so complicated? And if you're coming from a biology background, you say, why do you think the network is that simple? It has many, many more components that are missing in that. And in particular, the one in my lab that I studied for a long time is missing in that diagram. So why do you think that that's a good model? So the problem is those are both true. So you could but the idea is you have to, from a mechanistic point of view, you can't do everything all at once. You have to start, you have to build up some intuition, and then you can work your way back to something which includes more and more of the realism of real biological networks. So if you want to start small, you might as well start really small. So here is starting really small. This is the simplest sort of EMT network model for the bottom part here, for this decision-making network. It relies on having two transcription factors, snail and Zeb. These are, in some sense, the most common transcription factors that people see across cancers, across developmental systems that drive EMT. Upregulating these transcription factors then leads to all those downstream consequences for the actual behavior of the cell. And on the other side of the ledger, what we discovered when we read the literature was actually the thing which in many cases suppresses this, gives you this either-or behavior, or selects a state, were not actually transcription factors, were microRNAs. Now, I don't think anybody here has yet talked about microRNAs. I'm not going to talk about microRNAs because that's not what I want to talk about. But microRNAs are, again, regulation at a different level than transcription factors. Transcription factors regulate transcription. MicroRNAs regulate translation. MicroRNA is a small RNA molecule, 21 nucleotides or whatever, that goes and binds to messenger RNA, depending on base pair matching. And if it binds to the end of a messenger RNA, it interferes with that messenger RNA being translated into protein. So it's, again, a regulatory part. You can suppress snail by microRNA, and this suppression is taking place not at the level of the snail messenger RNA, it's taking place at the level of the snail protein. So you would have perhaps a relatively high level of snail messenger RNA, which you would see even in a transcriptome, but the connection between that and the level of protein would be modulated by this thing which is sort of interfering with translation. So in these models, the good news about this is that it's not meant to be biologically realistic in a deep sense. It's meant to be a learning example of what we can understand simply. And when you study these networks, you can figure out that there are different states, which we already knew, that there are states that are clearly epithelial, there are states that are clearly mesenchymal. And the other thing we discovered, of course, is that there are states that we call now hybrid states. So here is a picture of what a hybrid state looks like. from the point of view of the mathematics of this model, which I decided not to go through in detail because I want to get to what I really want to talk about in about 5 minutes or so. But if you look at this picture, what it says is if you vary something, so for example, this is the result you would get if you had an inducible snail construct in your cell. So if you had some way of inducing snail by adding a genetic element to your cells and you turned up the induction, what would you see? Well, at low levels of snail, if you don't induce it and the cell starts out as epithelial, it remains epithelial. So here you have epithelial cells. Then if you induce it to a very high level, you have mesenchymal cells, they're up here. And if you induce it to an intermediate level, what you get is a new possibility, something that we call a hybrid cell. It's written as E/M, at least the way we think about it. And the way you think about it biophysically is that if you look at its proteins, it's a cell that has maintained some amount of epithelial proteins and therefore epithelial function. For example, E-cadherin, which encodes for the stickiness of epithelial, making them form these nice epithelial layers, that is completely present. Its highest level is in epithelial cells. It gets completely suppressed in fully mesenchymal cells, and in these intermediate cells, it has some intermediate value. It's still there. You haven't gotten rid completely of these epithelial traits. But on the other hand, you've picked up at some medium level some mesenchymal traits. So if you look at the protein complement of those cells, and that's what we originally did, looking for these cells, you actually see something that looks like a mixture. You see levels of E-cadherin, you see levels of vimentin, you see levels of keratins, you see levels of, fibronectin. You see all sorts of things that you would never expect to find in a single cell, yet you find them in a single cell, and therefore it's a different state. It's a state that was not expected and then was verified in a number of experiments. And of course, the reason why this was important was what I described on Sunday, but just to remind you very quickly, well, maybe I should show one thing. So you can look for these things both in vitro and also in vivo. This is one experiment. This was just done from a sample of a patient sample, an early example of looking for these types of cells. This is from Itaiti Roche's group, who at the time was at the Broad Institute, and he looked at head and neck tumors, and in his labeling and his analysis of what was going on, if you look at the tumor, in the interior of a tumor, you typically see epithelial cells, because that's what they came from. These are carcinomas, so their original cells of origin were epithelial cells, and the tumor reflects that, the primary tumor in the center. At the edge of the primary tumor, you have cells that he's labeled partial EMT based on his signature of what contributes to partial EMT, one of which actually is this snail-2, which is sometimes known as slug, which is a very common thing which you see in a partial EMT cell, the transcription factor. And then he looked for full EMT, and he found very few tumor cells that were full EMT. Most of the cells that looked like they were full EMT were actually mesenchymal cells, were actually fibroblasts that were in the extracellular medium around the tumor. And so the picture that you have these different cell states, first you look to make sure that these exist at all, but then it turned out that even if you look at patient samples, that these things not only exist, but can be quite prevalent within the tumor. And then of course the reason why we cared was this graph I showed the other day, skip all that, the graph I showed the other day, which is experiments that started showing that, in fact, it's these partial EMT cells in the tumor that, of course, they can leave the tumor. They are motile. They can leave the tumor. And these are the ones that are most likely to create metastases. So I don't want to go through that entire story again. And it's very much connected to some of the stories about drug resistance that we heard from various people. And I just want to mention one more aspect of this. So I showed this picture on Sunday. I didn't show this picture, but it's a related paper, which is this picture, which is that from the time that the original experiment was done to now this is 2021, technology improved. We heard a very nice talk discussing the technology of lineage tracing and how it can be used for various things. And this paper used a very sophisticated method of lineage tracing, which is sort of the way, you should read this paper if you care about lineage tracing, because it really is a very interesting idea. You have a programmable lineage tracing system that as the system continues, you don't just rely on putting in your barcode at time 0 and then looking at some final point where that barcode wound up. At intermediate times up of your choosing, you can introduce mutations into the barcode, and you can track much more carefully how the cell's paths are diverging and eventually giving rise to what you see in the final endpoint of your study. So I recommend this paper for anybody interested in lineage tracing in the context of cancer. But from the point of view of my talk today, I just want to mention that they came to the same conclusion as was achieved in that earlier paper in 2018, which said that the most likely cells that could undergo this transition were the partial EMT cells. Okay, so that's my review of what I partially said on Sunday. And now let's move on. So I mentioned this experiment yesterday as a motivation for today's talk, and now I'm going to go through this experiment again. So this is an experiment that showed that, the good thing about biology is that problems never end. They just deepen somehow. So you work on something and you think you make some understanding and you seem to explain experiments and everything is great, and then you discover that there's a whole layer of things that you just don't understand yet, which makes fun life of working on things and doing new things, but it's sometimes you'd wish, okay, I've done that problem already, it's over, it's solved, let me go on and do something else. But the answer is EMT is not like that yet. So this is a paper about EMT again. This is done not with inducing snail, as in my nominal graph before, it's done by inducing a different transcription factor called twist. Okay, so twist is inducible in this particular experiment, and these are mammary cells that normally, of course, are epithelial, and then we're going to try to induce EMT. And what they did is they just did that. And this was around the era where people started to think seriously that maybe, single cells don't always go along with the bulk. You can have bulk measurements, which give you the average behavior. Most of the experiments up until this period of time, this is sort of paper written in 2020, which means the experiments were done maybe in 1998 or so. So up to that period of time, most of the data being used to explain EMT was bulk data. And, but around this time, people were getting better at developing live cell markers, and people were certainly getting better at doing single cell RNA-seq measurements. So it became clear that one should at least ask the question of, well, you've seen that the average of the population now, has expressed all these new proteins, all these new EMT transcription factors. How is that represented in individual cells? Are all individual cells doing the same thing, or is there some population level heterogeneity in what's going on? So this was an experiment that was attempting to look at this population level heterogeneity. And what they saw was, okay, here is a epithelial marker, EpCAM. They measured the initial distribution of EpCAM in the untransformed cells. Fine. It was very nicely, all the cells sort of fell into this, what they drew as an epithelial box for expression level of EpCAM. And then they did their induction, and here is the induction. This was done for quite a long period of time, 21 days of tamoxifen treatment that was coupled, where their induction element was coupled to an estrogen response element. And the technology doesn't matter, it's pretty standard by now, but what they saw is that most of the cells transferred to the box that they identified as being mesenchymal-like, low expression of this epithelial marker, and some of them, went partially there and then sort of stopped. So these cells here had some type of arrested EMT. Now, again, this was, before the sort of general realization in the cancer community about partial EMT cells. So they had to label things as either E or M. They didn't have any other choice. So they called these things mesenchymal. If you actually look at it, what you see is they're sort of in the middle. And if you actually look more generally at these cells, which they presented other data of other proteins, other genes, they're actually hybrid cells. They're actually did not stay completely epithelial. These cells actually underwent a transition to something like a hybrid cell. Now that's completely consistent with all these experiments that I told you about that said that it's the hybrid cells that are most likely to generate metastases, because they showed in their paper that if you took those cells and only those cells and injected them into mice, those were the ones that could generate new tumors efficiently. So it all held together, all nice, but the question arose then, okay, fine. what was the difference between the cells that went there and the cells that went here? Why did some cells stop halfway down the path? Now, this is an incredibly important question, because if you think the ones that stop are the interesting ones, you really want to know what made them stop, or what was the bias in their initial state that that convinced them that they should only undergo a partial transformation. So, as I said yesterday, they didn't answer the question in this paper, but they pointed in the direction that I think is eventually the right direction. The direction they pointed in was this statement in their paper that the ability to retain epithelial traits, that's what this means, to go to partial EMT where you still retain your epithelial traits, was connected to some type of epigenetic program. Epigenetic program here, as we'll discuss in a moment, just means that it's another layer of regulation of what's going on, having to do with at the same constant level of some transcription factor. It may or may not be effective at generating its downstream effect if you make the piece of DNA that it's trying to bind to, if you make that very accessible, or you make that very inaccessible. So how do you make DNA accessible and DNA inaccessible? Well, DNA is this long thing. It wraps itself up in various complicated ways, and it has nucleosomes. And the issue of accessibility has to do with how you arrange markings, chemical markings along the DNA of a whole variety of types. And some of those markings create chromatin that's very inaccessible. It's very hard to bind to those elements. And some of those marks create regions of the chromatin that are easier to access. And they argued that this phenomena, this epigenetic layer of regulation, was the thing which was determining the difference between the ones that were able to undergo the full transition and the ones that were only able to undergo the partial transition. And their evidence, and they didn't, as I said, they didn't really provide any real data about how to think about that. They just said, well, we can think that that's the case because they looked at, one of the things that this Zeb factor does, this is the factor here, one of the things that does is it binds to the promoter regions of epithelial cells and suppresses them. So if the promoter region of epithelial genes, if the epithelial genes become less open, less available for binding of the Zeb transcription factor, then the system will be less efficient or unable to downregulate the epithelial genes, all else being equal. You might have the same level of the Zeb transcription factor, but because your DNA is less accessible, you'll get an inefficient downregulation of the epithelial factors, and that's what was just seen experimentally. So this was a hypothesis that they really didn't provide any mechanistic framework for testing or evaluating. It was just an idea. Ideas are good, but you got to go beyond them. So we've been thinking about that idea at the time, and then I don't need to walk over there. I have this nice button. So I want to mention just one other motivation, which is sort of why we were thinking about this question, and it's a sort of two-way street. In other words, just like you can go from epithelial to mesenchymal, but you don't always do it, you're something that can hold you back, you can also go back. I mean, you can go from mesenchymal to epithelial. It's, on many conditions, this is a reversible thing that happens. We heard that the transitions don't have to occur the same pathway. You can go back, you can come one way like this and go the other way like that if you have some two-dimensional plot of the pathway, which we saw yesterday. But this experiment is about the lack of reversion from mesenchymal to epithelial. So let me describe this experiment, because it really did motivate a lot of our thinking about this question. This is a mouse melanoma experiment. And these people did the following, they're looking at some models of mouse melanoma, and it's good because you keep imaging what's going on. It's you have a tumor growing in the skin here. So here is their cartoon of this tumor. This is a paper in 2015. And what you see is the primary tumor growing and growing. And then what they discovered up here, you see they colored it in red here. I really like cartoons in cancer biology papers, because they're really very good at showing you what they think is going on. I really am impressed by their whatever graphic artists they hire to do these things. We're so crappy at doing this in our group that it's pathetic. Anyway, so here they have this red region. It turns out what this red region are concentrations which they measured of the binding partners of the Notch receptor. So there's Notch ligands. Now, you don't have to know all about the Notch system. It's a very well-studied system in a whole bunch of developmental contexts. Notch is a receptor on the surface of cells. It has ligands which are called delta or jagged, many, different types of deltas, different types of jaggeds. And if you go back, I'm not going to, here, to my picture, you see that in the real world, if you want to figure out how to activate, let's say in the lab, or let's say in the real cell, or the real body, EMT, you need some input that comes in that does that. In the lab, they usually use TGF-beta, but Notch will also induce EMT. So the Notch signaling system, if it's activated, will induce EMT. So their logic was that the primary tumor reaches this layer in the skin up there. That layer happens to have a different chemical microenvironment, which means it's rich in these Notch ligands. Those activate the Notch receptors on the cells that are growing as this melanoma, and that induces a motility transition. We don't tend to call it EMT in melanoma because melanoma cells are not, melanocytes are not really E cells, but that's a technical detail. It induces exactly the same type of transition as you call EMT for epithelial cells. And so then their picture is, well, all of a sudden these cells have become motile and they leave this epithelial region and they start migrating throughout the rest of the body. Now, this is a great picture because number one, it illustrates one of the most important things about metastasis that needs to be understood in the field of metastasis from my perspective, which is there are obviously genetic correlates to being able to metastasize. Everything, all these networks, the parameters in them, which ones couplings are there, which ones are suppressed, all depend on the genetic background of the cell. So mutations clearly can bias certain clones to be more metastatic than other clones by exactly how they set up these networks. However, this is a perfect example of showing that a new genetic mutation that is a metastatic gene is not necessary. Why? Because here, nothing happened genetically in this experiment. The cells, the primary tumor just grew, and EMT was induced by a purely microenvironmental change in the chemistry, and that was enough to cause the cells to metastasize, to leave the primary tumor, go elsewhere in the body. So genetic changes, mutations, clearly are biasing what the network is doing. We all know that. And the normal cells in the skin are not growing out of control and metastasizing. That's just not happening. So obviously, whatever mutations there were in this particular melanoma line, BRAF or whatever, was essential in creating the tumor and essential in allowing this cell fate transition to occur. But there's a difference between allowing and causing. I think that's probably so. This is a great experiment for sort of making that point that I think is quite important to realize from just the general field of metastasis. But anyway, so that's why I like this. That's why I noticed this paper and always put it in my talks. But it also asks the same question in some sense as that other paper, and unless they didn't ask the question, but I asked that when I read the paper. I asked, well, look at the picture. What happens is all of a sudden these green cells have left this red region. So they're no longer subject to this signal. All of a sudden, they used to have a large pro-EMT signal impinging on their network. They've switched. Now they've left that region. Why don't they switch back? Why all of a sudden do these cells stay mesenchymal? Well, obviously there must be some very large hysteresis. There must be some very large reason why the cells, even for a long period of time, we're not talking about hours, we're talking about days, weeks, whatever, don't just revert back to epithelial when they're back in their normal microenvironment. So you need an idea, you need some concept as to what could prevent MET in some cells. And the other experiment asks, under some simpler conditions, what could prevent EMT, or at least complete EMT. Okay. So onward. So this brings us to a different experiment. So let me tell you about this experiment. So Yun Zhang was a postdoc in Bob Weinberg's lab. Now he has his own lab in Beijing. And he gave a talk in 2020 at an American Association of Cancer Research meeting. And of course, in 2020, no one gave in-person talks anywhere under any circumstance. So of course, he gave the talk on Zoom, which made it very convenient for my student, particular Shaon, Chakrabarti, to see the talk because, he was sitting there and, there was no group meetings or whatever. So he was just sort of looking through talks on American Association of Cancer Research meetings. And he came across a talk by Yun Zhang. So this was 2020. It took till 2022 to publish the data. Now, it's very interesting if you contact an experimental biologist and you say, your talk has been really interesting, can we work on your data? It's not always the case that they're willing to give you the data before their paper is published. Just, for whatever reason. I mean, it's good sociology, bad sociology. I don't know what they're worried about, but nonetheless, they somehow feel like, you're going to somehow steal the limelight or something. And I think it's exactly the opposite. From my point of view, if you have a better analysis of your data, it actually helps you get your point across more, not steal some part. But none. So he had none of that. He agreed immediately to share all his data. So what experiment did he do? He did an experiment which was quite analogous to the one by Christina Shaon that I described already. He took, again, cells that were very similar. These are, again, this sort of HMLE type cells, so it's a little bit different. This also has a RAS oncogene in the cells, so it doesn't matter. There are always details. And again, he tried to induce EMT by TGF-beta treatment, so it was not a genetic induction, it was a pharmacological induction. Again, an in vitro experiment, all fine. And what he again found, and this is now a UMAP, which we heard again all about yesterday, so I don't need to go over what UMAPs are and how you use them to create more, let's say, interpretable representations of high-dimensional data. So here is the UMAP, and what he saw again was that there were two subpopulations. Now they were much more equal in size than in Christina Shaon's experiment, but okay, details are details. And also in this case, one of these, this one, underwent what looked like complete EMT, and the other one underwent no EMT. So rather than partial versus all, here it was sort of all or nothing. It really was, these ones went to something that looked pretty mesenchymal. Here is, what these things look like. This is one set of cells here where you look at various levels of various things. This is looking at CD44, which is a marker of mesenchymal cells. It's a surface marker, so it's easy to measure. And here you got nothing, essentially. And the control was nothing. Giving TGF was nothing. Giving a TGF inhibitor was nothing. And this one did exactly what you thought EMT should be. It had some low level control, and then you raised up by some significant level when you gave a TGF-beta, and it went back to sort of the control level when you gave an inhibitor of TGF-beta. So again, the population behaved in two distinct ways. One was very able, willing, quick to adopt the EMT lifestyle, where you gave it the signal and it said, yeah, I'm going. And the other one was just reticent. It could not go, it could not make the transition. And what Yun Zhang did was something which was different than what was done before. He didn't just characterize this, he tried to actively change what was going on. So what he did was he created a CRISPR screen to look for things that could be knocked out via CRISPR that would change the reticent ones into ones that would undergo EMT. Okay, so he did this entire CRISPR screen and again checked which ones could go EMT, and he found a number of hits, of course. The two leading hits were two enzymes whose role is it to modulate the epigenetics of the cell. So these are enzymes, one of which, and I'll talk about these in a moment, is a part of the PRC2 complex. So this is a complex that's involved in putting inhibitory marks on histones. Histones are these, little spool-like things that help organize the genome. And this thing puts a mark, which if you want to read the epigenetics literature, you need to learn this language of all these marks. This is H3K27ME3. That stands for histone number 3, lysine number 27, adding 3 methyl groups to that lysine. Okay, that's what H3K27ME3 stands for. It's a known to be inhibitory mark. It tends to prevent the activation of the gene. And the other enzyme we found was KMT2D. Again, the language is, this is methyltransferase. This is adding a methyl group again. But this methyltransferase adds it to H3K4 instead of H3K27. And H3, the fourth lysine, when you add that methyl group, then it actually serves as an activating mark. Now, you can put on your molecular biophysicist hat and you can try to explain in detail why is it that adding a methyl group to the 27th lysine does one thing and adding methyl groups to the 4th lysine does something else. We're not going to do that. I'm not, that's a perfectly fine biophysics problem to work out at the molecular level, understanding how these different methyl groups change the charge on the histone tails and how it determines wrappings of the histones and ability of the DNA to access, the ability of the transcriptions to access those things, the ability to remove nucleosomes as you actually elongate to create your RNA. All that is perfectly fine. We're just going to take as given experimentally that there are these marks created by one of these enzymes that's an activating mark, and the opposite mark is an inhibiting mark. And the other thing to keep in mind is that if these things mutually repress each other, you can't have on the same histone and on the same nucleosome, you can't have both activation and inhibition. You either can have no marks whatsoever, that would be a sort of null state, a control state. You can have an activated promoter region, and you can have a inhibited promoter region. So you can think of there being 3 states for the promoter region of various genes, and it turns out that ZEB1, this one, is one of the genes that is directly acted upon by these particular enzymes in its promoter region. So the expression level of ZEB1 is being modulated by whether its promoter region DNA has a null mark, it has no marks, whether it has an activating mark and has an inhibiting mark. Okay, yes. Yes. I don't remember how, the truth is I don't remember how long he did that for, but EMT takes at most a week and something, so I'm sure he did it for like a couple of weeks. And it wasn't a question of just going slower. He definitely checked that it wasn't just that they did the same thing, just it took longer to do it. They were really reticent. At least up to some, he didn't wait 3 months, but certainly on the normal timescales of EMT, which 3 days, 5 days, whatever, it was, so you look double that time, triple that time, and they just don't transition. So I don't think we have an answer of if you wait forever, that's a good question in a sense, because if you're arguing that it's the epigenetic marks that are somehow responsible on some time scale, you can have spontaneous changes of your epigenetic marks, and those could then put you in the other category. But he didn't wait long enough to see whether that's going to happen. If you wait long enough, we don't know. But how do you separate these populations? Well, CD44 those are just done by usual UMAP of single cell RNA-seq. Oh, just the two clusters you see. Yeah, no, really, he didn't separate it. Well, no, he did separate. No, I shouldn't say that. He did separate it, but CD4 was a good enough marker. If you look at this graph over here, if you have something which couples to fluorescent CD44, this one has nothing, and this one has a large value. So it's pretty easy to separate, I think. As usual, there's an infinite number of experimental details. That, yeah. That was the claim. That was the claim. Yeah, so you could separate, and then you can check, and yeah, that was the claim. Yes. 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 Yeah, oh, sure. Yeah, of course. There's an, I didn't put them in the graph because those ones didn't, were not the ones that were identified in this screen. But yeah, there certainly is some enzyme here that goes back to here, and I don't remember the name of it. So these are methyltransferase. There are demethylases. I don't remember the exact number of the, so there's a KDM something that does this, and I don't even remember which one it is. And there's something that does that. So yeah, no, these are all enzymatic reactions. So yeah, I didn't mean to imply there wasn't anything there. Okay. Now, the reason why we noted, I mean, besides the fact of our general interest and the fact that we were thinking about this problem, there was a specific reason why we generally, sort of, I wouldn't say we dropped everything we were doing and tried to explain this, but, it's somewhere almost there. Because this paper had an extremely bizarre part of what they found. And the extremely bizarre part of what they found, we think, provides a hint, or at least a clue, as to how one has to begin thinking about creating models of this type of epigenetic coupling to the genetic networks that we've all been hearing about. So the experiment looked like the following. So I said you knock out PRC2 and you knock out this KMT2D, and in both cases you get some type of EMT. Now it turns out that when you knock out PRC2, this is knocking out the inhibitor, okay? You get something that looks like partial EMT. You get an intermediate level of ZEB, and here are just some examples of genes. You get an intermediate level of ZEB, and you get some level left over still of EpCAMs. You get something that you can identify based on either direct, things like this. I think someone out there showed Western blots at some point, or you can look at the single cell RNA-seq. You get something which you can identify as a partial EMT state. I should say just technically that when you gave these inhibited, you didn't even need to give TGF-beta. They would spontaneously go into these states without actually inducing them. So you didn't have to actually induce them. They were already, at baseline, went into these EMT states. Okay, so loss of PRC2, loss of the inhibitor, gave you something which looked like a partial EMT state. Loss of KMT2D, loss of the activator, gave you something that looked like a full EMT state. So let me repeat that because it's highly non-trivial, right? You knock out an inhibitor and you expect the level of various things like the EMT transcription factors. And I said we already knew based on ChIP-seq data and whatever that ZEB is directly a target of these particular epigenetic modifiers. But there are other ones that are also relevant, but that one we knew for sure. So you knock out something that's supposed to be an inhibitor and you get the response you expect. The ZEB level goes up. And then separately, you go back to the baseline and you knock out an activator and you expect the baseline level was 0, so you expect to see nothing. It was sort of the control. And in fact, it went up even higher when you knock out the activator than when you knock out the inhibitor. So this tells us, first of all, that, so the first thing it tells you is that unlike the previous experiment where there was only sort of circumstantial evidence that manipulating the epigenetics could feed back directly into the EMT system, here you had direct evidence. You can manipulate the epigenetics of various genes, and you could directly see the consequences. So that's already good because we now had much better data. And we, of course, had single-cell RNA-seq for all these different possibilities. But it also presented a puzzle, like, why does this work? What is this telling us about epigenetic regulation that's interfering with our intuition that we had about what should happen? Now, people know, various people, even at this meeting, but always, sort of ask me, how I decide what to work on. Usually students want to know, well, I work on a number of different things. As I'm sure you've noticed, I work on immune system things, I work on EMT, I work on cell motility, I work on a number of different things. And people want to know, well, how do you figure out what to work on? So I usually say, for me, it's actually pretty simple. You look at papers and people present talks and you read papers and students tell you about papers. And before hearing what the conclusions of the paper is, I try to form in my mental mind a guess as to what the answers are going to be. Someone did some experiment, I try to guess what the experiment is going to show. And, hopefully I'm right a significant part of the time, because I've been working on this for a while. But every once in a while, and this is an example, you just guess completely wrong. And so the experiment does something, gives you a result, which is completely counter to your intuition. So that's a good demonstration that there's something missing in your intuition, right? If your intuition were perfect, you would have said, of course this is true. I know everything about how epigenetic regulation works, and of course this is going to work. But the answer was, I didn't know how epigenetic regulation was going to work. So in fact, I didn't know why this was true. So we created a model which to try to explain this feature of the data, which to try to create a coupling between the genetic network, something, a small-scale genetic network. It's not this one exactly, one that's more relevant specifically for this cell line, which has its, It has actually a few other EMT factors that are important, but sort of this general category, and couple that to some type of epigenetic regulatory mechanism. So that's what we did. By the way, yes. Yes. And many other things, which is going to be the point I talk about now. Yes. 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 So the answer is this three-state behavior is relevant for a number of the EMT transcription factors that are driving EMT in this system, as seen by RNA-seq and other previously known experiments. Okay. Say again. Here, no, not yet. Right, this was our original, this is what our genetic network works like, and now I have to tell you how we're going to modify that network to include some type of epigenetic effects, which is very rare. I mean, people have studied epigenetic regulation in isolation, just what things control the marks that go there and how things are coupled. People study genetic networks in great detail, which we've been hear about, and very few people have tried to put them together in one network. Now, if you want to see the of course, I'm going to give you a very general view of what we did. If you want to see the details, this is described in a paper about, I don't know, a year ago at this point in PNAS, where the first author is Al-Radawi. So it's PNAS. If you search for me and you search for the word epigenetic, you'll find this paper. So all the details are in a very extensive supplemental information for those who like equations and formal discussions. All of that is in the supplemental information of that PNAS paper, and all the results are in the main text, as is usual for a PNAS paper. By the way, in PNAS, the editorial board, they often say the rule is, and apparently this was actually published there, that you lose 10% of your readership for every equation in your main text. so what? No, the SI is fine because the SI is only going to be looked at by people who actually care about equations. So but the point is, if you put it in the main text, there's some, it's like a gate. You reach the first equation, and 90% of the people get through it, and then you reach the second equation, and okay. So if you put too many equations, by the time you reach, no one has reached the end, except for some die-hard computational biology people. So anyway, the PIS paper is written in that framework, right? The ideas are tried to describe in words, like I will try to do here, and then all the details of the equations are sitting in the supplement. Okay. So what is our picture going to be? So I have to tell you how this thing is going to work. Let's see, is there another thing? Oh yeah, so I'll come back. Okay, I can do this in 2 stages. So what we looked at in this system is we looked at some mechanism that sort of looks like the following. So we have these enzymes, and these enzymes are responsible for placing marks on the promoter region. So let's say we have some promoter region here. So on the right is going to be the ZEB promoter, and it's going to be coupled to this type of structure. And the type of structure has sort of 3 dynamical variables. It has the level of P, the level of T. P is the repressor, and then there's an activator, and actually it turns out there's also another activator, KMT2C, which is relevant in this story. Okay, now, what is the dynamics that you're going to put into this model? The dynamics you're going to put into this model is the fact that in order for one of these things to create a mark, what it has to do is it comes and binds to the region, to the promoter region. It creates the mark. It stays there for a while, and then it leaves, and the mark can stay there stays there afterwards. So there's a percentage of the time when the mark is isolated by itself, but there's a significant percentage of the time when the mark is associated with actually physically having those enzymatic factors attached to the DNA. Now, imagine that you have a system where you do not have an infinite number of these enzymes at your disposal, right? So you can look up the numbers, and there are many, there are 400 sites that are subject to this regulation, 400 places in the genome where these enzymes can couple. You have a limited number of these enzymes. So your enzymes are wandering around, binding to various regions. And of course, if they're bound to some subset of regions, they aren't available to bind to other regions. So there's a dilution effect here. The dilution effect is whenever you have different competing sinks for something, you can't satisfy all those sinks at the same time. You can't be in place A and place B at the same time. So if you have a limited number of these enzymes, you have to take into account not just the site you're looking at. You only care about the ZEB promoter or maybe a few promoters. The problem is those enzymes care about 500 sites. So they're not going to only obey the equations you would write down for your one site and say, oh, I have some background concentration of these enzymes, and I can figure out based on some simple binding equation, binding kinetics, how much is attached and how much is not attached. What the background level is depends on all the other sites that you're dealing with. Now, you can write down an equation for 500 sites, but that's not very good, to try to make progress. So what we did is what in physics would be called a mean field approximation. A mean field approximation is, we said, there's the sites you care about, 1, 2, 3, whatever, and the rest of it is some background, but it's not a constant background of enzymes, it's a constant background of a genome. It's a super genome, super element that has a lot more sites, so it is able to attract many, many more of these binding erasers and writers, and you have this competition, which you can write as the few things you care about coupled to interacting with this pool of things you don't care about, but which can play an important dynamic role. Now, why is that important? Why is that important? So let's try to understand the question of what happens, why is it possible, forget about the details, but why is it possible to knock out an activator and get an increase in the expression of your gene? That's the first question you can address with some thought process like this. And the way that works is you would say, well, the level of your 3 states in your system is controlled not only by the enzymatic concentrations you put in, but it's coupled to what are your interactions with the rest of the world out there. So the rest of the world out there can basically have different affinities to these different connections compared to you. So under the normal condition in your cell, you can have a cell which has PRC2 bound to your system. It's inhibiting your gene, but on average, the other genes in the system, on average, because we're talking about 500 other genes, on average, those ones are going to be more, have, more KMT2D binding affinity. They're going to attract all the activating enzyme. You're going to be inhibited. You've attracted the inhibiting enzyme, and they've attracted the activating enzyme. Now you knock out the activating enzyme. What happens? Well, all of a sudden you have this huge sink out there that no longer has the activating enzyme to block its ability to soak up the inhibiting enzyme. So what does it do? It takes your inhibiting enzyme, removes it from you, and puts it elsewhere in the genome. So the effect of knocking out the activator was removing the inhibitor from what you care about. Okay, so let me say that again, because that's the key insight as to why you can get non-intuitive results. Because you have this competition, which you have to evaluate not the absolute level of what you're doing. That absolute level, of course, you no longer have this activator, but the idea is your gene started out being inhibited, you were in the inhibited state, and knocking out the activator can actually remove the inhibitor for you because now the inhibitor can be sucked off, diluted by all the rest of the genome. Now, of course, that only occurs based on certain parameters in the model. You have to arrange the interactions, right? I mean, this is not a generic property of that type of picture. You can arrange, you have to pick, the way in which things couple to different ones. And so if you look at that, I have this, this is the same, whoops, let's go to the next one. No, doesn't go to the next one. Try again. Okay, so if you look at this, you can say, well, how likely is that to happen? In other words, is this really possible that, that the EMT factors were in this strange position where you had this result happen? So you can look more generally, genome-wide. So here is, I said, 413 targets that were known to have this regulatory motif important in it. And for those particular ones, you can ask a very simple question. You can say, I have my baseline cells. You take my cells. Then you have the ones where you knocked out the activator, and you have the ones where you knocked out the inhibitor. And you can say, if I look at the level of that particular target, which one what is the ordering of strengths? Is it highest in the baseline? Is it highest when you knock out the inhibitor? Is it highest when you knock out the activator? What's the distribution of those effects? Well, here's the graph done by looking at all those different things in the RNA-seq data. So it turns out that by knocking out the repressor, most genes behave in a normal way. You knock out the repressor, and the highest level you get of the gene expression is when you knock out the repressor. Okay, that's the intuition. So intuition is true in 67% of the genes in this particular system. And other cases, there are 11% of the case where you have even a different thing happen. The control level is the highest expression. So knocking out the activator or the inhibitor lowers it in both cases. And in the third case, it's the one that seems to apply to the ZEB promoter and also another transcription factor promoter in this system. And this is the case where the highest level is where you knock out the activator. So it's not very rare, it's 15%. Why is the EMT system in that category? Who the hell knows? I didn't design the system, so I don't know why that was the logical thing to do. But this way of thinking about it can help you begin to explain how you can not only modulate these effects by changing the accessibility, but you can also then, begin to explain how sometimes you can get very counterintuitive results via this dilution mechanism. Okay, now, once you have this idea, then you can go and do the details. Does this go? Yeah. Yes. Yeah, so this shows just those different cells that are in these different categories. Oh, those are the parameters of the system. So those are the parameters of the model. So global, local A, B, these are how much these different enzymes are attracted to local things versus the global average, the mean field. So those are just parameters in this pretty complicated model, which you could, yeah. Yeah, the CDH2 is N-cadherin. So we just looked at various genes and mapped them on, so for various genes, since we know the answer, we could map them onto which of these categories they fell into, and then you could postulate how this would work. So the way this diagram works, I didn't go through this in detail, the way this diagram works, this is just a sort of pictorial representation of the mechanism I told you. This is the baseline. So in this baseline, you have a repressed gene over here. So all these three things were here. If you knock out the repressor, in this case, you don't have a strong attraction to the mean field here, so you still stay repressed. And if you knock out the activator, then, I should say differently. If you knock out the repressor, then this thing goes up a little bit by a lot, and here this one goes up the maximum if you knock out the activator. So this is not the case of ZEB. The case of ZEB is, I guess, the top one here, twist, which is another EMT factor, where this thing is repressed. So this repressor link down here is strongest. You knock out the repressor, you get nothing here. You get a sort of null state here. And then if you knock out the activator, then what happens is this repressor gets sucked off into this part of the genome, and now you get a strong activation. So this is just a pictorial representation of what I tried to explain in words. It may help or may not, but anyway. So mean field means that what our model looks like is we have a few genes that we care about. SEB we care about. I'll show you you can see on the next slide which genes we care about. So the model consists of ZEB1, and actually there's a more complicated full model. This is the full model. So this is what our genetic network looks like at the end of the day. Again, at this scale, but done specifically for this cell model. So here we have snail, but there's also another transcription factor called PRRX1, and there's also the microRNA regulation. So this is our local genetic network. That's what it looks like. And these two genes, ZEB1 and PRRX1, are both subject to this epigenetic regulation. So our model looks like we have the ZEB site, and the values of what the state variables of the ZEB site are null, activated, repressed. We have this site, and then we have the average site, which is a much bigger set of sites with an average value, activated, suppressed, null, repressed. And then we have enzymatic concentrations, which change. It changes because as you if you want to write and read something, you have to bind to that region and stay there for some amount of time. And if you then have those as dynamic variables, you can figure out what are the populations of your various receptors, your various promoters of the genes you care about, and also what the average behavior of the rest of the genome is. So that's what the model looks like. And the details are long, and so I'm going to leave them for reading the supplement for those of you who get past equations. So, okay. So this model, of course, as usual, you wouldn't talk about it if it didn't work. So this model can explain, it's built to explain the most counterintuitive parts of the data. That's where the model was created to do. But then you can quantitatively go and fit the data for various genes, and there's no problem anymore. Once you've gotten the right possible behaviors, you can adjust the coefficients and go through the exercise of figuring out the right coefficients of these things and fit the experimental data. So that's that's simple once you have the general framework. Yes? Yes. Yes. That's exactly what happens. Well, you're giving a meaning to the facts, right? You're saying as a means of compensating. I don't know if it's a means for compensating, it's just what happens. Right? So what you said is exactly right, that if you knock out the activator, the repressor gets sucked off the genes that you cared about and gets sent to the rest of the genome where before it had no room to bind. Whether that's on purpose, whether there's some design reason why you'd want the ZEB promoter to have that structure, I don't know. Maybe if you think of some reason, please let me know. I'm just saying this is what can explain how this works in these. But the thing that I want to, okay, it's huge. So let's put it this way. The issue is not, from my point of view, to explain a particular data set, right? We can do that. That's your test that what you're saying makes sense, is you can explain particular data for this particular system with a model which has a reasonable number of parameters. So it's not like it's impossible. But the point is that models that can explain data sets have to have built into them the possible mechanisms that are at play in that data set. If you don't build in this competition, you will never get an effect that says you knock out an activator and the level goes up. It just, by definition, it won't happen. So therefore, it isn't a question of fitting the parameters of the model. Once you have the right mechanistic framework, you can fit the parameters of the model and compare it to the data and do however you want to do that. It's not even that hard in this case because a model only has a few genes and a few things. And so we can do that. It's an exercise that's useful because it's good that you can do it, but that's not the point. The point is that you learn something about what the possible mechanisms that are going on in a system like this by going through the ability to be able to fit the data and see if you can make sense of what's going on. That's really the point. And then since you learn about that mechanism will be slightly different in other circumstances, but it told us some new lesson about how to think about epigenetics coupled to gene networks. So let me finish with a couple more quick things related to this. Now, as I said, biology never ends. And this is Mark 1 of the model, where you notice that this is a feed-forward architecture in some language. A feed-forward architecture means that the epigenetics are affecting the genetics, and there's no feedback. My principle, I have a number of principles about biological systems. The first principle is, you always have to have 10 molecules when one would do to make life hard. And the principle number 2 is there's no system that doesn't have feedback. So clearly, this is only the first level of thinking about the problem because it turns out there are a variety of known cases, known biological ways in which the genetic network can feed back to the epigenetic network. So here's one. So it's known, for example, that in this paper from 2021, that if in this particular system, as RNA is transcribed, so as the gene is actively being transcribed, it can actually bind to some of these epigenetic regulatory factors and therefore interfere with that regulation. So this is a feedback mechanism. You can't solve the problem anymore by first solving the epigenetic problem, using that to modulate your transcriptional rates, and then solving the genetic problem. Now you have to solve the problem as one big whole thing. So it makes your life harder, but it's much closer to reality. But the benefit for making the problem harder and having feedback is now you can have interesting things happen, which won't happen if you did a purely feed-forward model. So what's an interesting thing that can happen? Well, here is now the fuller model. This is the model Mark II, I would say, where you have this promoter circuits coupled to these two genes. You have coupling this way, but the level of those gene expressions can now go back in and modulate the system. And the advantage of having this type of feedback is, I'll show you on the next graph, is now you can actually get hysteresis effects and different states of the system, and you can make new predictions. So what you can predict in this system is that now if Zeb can feed back onto this epigenetic regulation, you can have a self-consistent Zeb high state, and you can have a self-consistent Zeb low state. In the Zeb high state, it's biasing its epigenetic feedback to allow it to be on, and in the Zeb low state, it's biasing its epigenetic feedback that keeps it low. And those states are accessible depending on the initial conditions in your system. So if you start out, so the prediction you can make, and this is currently being tested, is that if you do serial knockouts, if you knock out both the activator and the inhibitor, but you knock them out in different orders, you actually go to different states of this combined model. So in one case, you knock out something, you start out with some value of Zeb, then you knock out the other one, and you can change the value of Zeb that you go to because you can be moving a different way through this now more complicated states. So this still needs to be worked out in detail. I said there are a number of predictions you can make. Some of them were already tested in this model. For example, I told you that we had a model that had 3 epigenetic factors. We had PRC2, we had KMT2D and KMT2C, and the one that was seen experimentally was 2D, and it turns out in our model, one can ask, what would happen if one knocked out, so you can fit the model to all the data, whatever, and then you can ask what happens if they would have knocked out 2C instead of 2D, and the answer is nothing happens. It just turns out that in this model, with the parameters you get, the 2D is the one that's affecting really what's going on if it's there. If it's not there, then you need something that helps you activate the system in the absence of the inhibitory marks. And it turns out, so it has a large effect on fitting the data with the activator knockout. But if you still have the original activator there, knocking the alternate activator out has no effect. And that was actually seen experimentally. So experimentally, they had this whole CRISPR knockout table. One of the things they did knock out is KMT2C, and they saw no important effect. So that was a prediction. It's a post-diction in the sense that the result was in the data, but it was, but we didn't know that. It was, because they didn't point that out, because why would they list all the things that didn't change the phenotype in their paper? But if you went and asked them and said, well, what about, can we knock out KMT2C? They said, well, you already did that, and we know it doesn't change anything. So it's sort of confirmation. It's better to have a more striking prediction like this. And so this serial knockouts in different orders is hopefully they'll be doing this experiment now that Yun Zhang has his own lab in Beijing. Now, other predictions, about partial knockouts are also non-trivial. There's a very big difference in what happens with partial knockouts versus full knockouts for these different things. You can predict all these things, and hopefully these will be tested. I want to mention one last thing, which is, in some sense, I don't know, it's not corroboration of the details of the model, but it's corroboration of the general idea that this type of behavior is what's at least important in governing EMT. So this is a totally different system. This is now pure RNA-seq measurements of EMT cells undergoing transition. This was an experiment that we collaborated with in the group of Mani at the time at MD Anderson. And it's just very straightforward, but doing straightforward experiments when the technology is first available is good because you get to be first or almost first. So they just did, time courses of normal cells. This is MCF10A for those of you who care, undergoing EMT via TGF-beta. And what they saw again was, single-cell RNA-seq showed different trajectories. There were 3 different trajectories. You can see these 3 different trajectories in this picture. Whoops, you could see it if I didn't kill the graph. Well, this is not good. Okay. This is all yesterday's talk. I don't want to do that. You can see the talk go by quickly again. Slow down. Oh, went too far. Okay, so this is just reconstruction of single-cell lineages based purely on RNA-seq. This uses a method which was developed by numbers of people in the cancer biology community. I guess the most famous development was by Aviv Regev's group. And, it's basically a method. The method's called optimal transport, where you're able to figure out if you see the data at various time slots, you can use this method to try to figure out how the population distribution at, let's say, time after the EMT transition has occurred, how that related to the population distribution, to the distribution of different genes at the initial stage before you gave it the inducer. So you can sort of try to run time backwards using this method and ask the question of, in this case, they again saw, this is now no perturbations, nothing else, a different cell line, a different protocol, and here they saw all three possible fates. They saw cells that did not undergo EMT at all, cells that underwent partial EMT and stopped, and cells that underwent full EMT. And using this method, this bioinformatics method, you can go back and ask, what were the signatures of those cells at time zero that biased you to undergo one of those three possible fates? And in particular, we were interested again in what was the signature that biased you to undergo the partial EMT cell fate. And the answer was consistent with the CRISPR experiment in the sense that I told you that the CRISPR experiment, if you knock out the inhibitor, is the one that goes is the one that goes to the partial EMT state. And one of the leading things that was seen in trying to reconstruct at time zero what was going on that biased you towards the intermediate cell fate was low levels of expression of PRC2 complex components. So it's consistent. I mean, it's not a test, it's a different thing, but here there's no knockouts, there's no perturbation, there's just trying to say, can you use that information to see how maybe the natural epigenetic state of the cell is biasing what happens once you provide the perturbation? And the answer is what you get is pretty consistent with what we saw in the other experiment. This is sitting on the bioarchive somewhere, trying to get published, the usual detail. Okay, this is very nice. And of course, you could do this also with lineage tracing, which would be nice if somebody did it with a more direct method of doing this, but this is what we did. This experiment was done at this point, 5 years ago. We didn't have these fancy lineage tracing things, so we just had simple, at that point, rather novel single RNA-seq measurements. Now it's, every lab does this. Okay, and this was done, actually this is done by Yu Chen, who is a postdoc in Franziska Michor's lab. This is another great advance. So this whole project somehow revolves around life during COVID, because I already told you about the initial start of this project where my student found this movie, and we interacted for 2 years by Zoom with Bob Weinberg's lab, even though his lab is approximately a 20-minute walk from my office. You cross the river, what the geography of Boston is. Northeastern is on the Boston side of the river. MIT is immediately on the Cambridge side of the river. It's literally like a 20-minute walk to MIT from my office, and yet we never met this person. I still haven't met this person. He moved to Beijing somewhere along the way, and we've collaborated with him now for 3 years, and I still haven't met him. So the Yu Chen is a postdoc who arrived at Dana-Farber in Franziska Michor's lab. Franziska is going to give a talk next week, I guess, online here. He arrived there in April of 2020. And he didn't meet any of his lab mates or his boss, Franziska, for 2 years after that. And he started sort of getting crazed about this at some point. After a year went by, he just needed people to talk to. And we were the closest group that he could come and talk to, we're only, again, a 15-minute walk away from Dana-Farber Cancer Center. So he started, and I met him in some meeting somewhere. So he started saying, can I start coming? Can I start going? We started having in-person group meetings again. And so he started coming to our group meetings and started talking to us, and eventually led to this joint work. And he was interested in these data processing techniques. And so we eventually led to this work, which again has a lot to do with the COVID being at least having some positive benefits. Okay, so I'm done. Here's a picture of some of the people. Okay, so these are the lessons. You can manipulate the EMT process that will bias you towards either not doing anything, undergo full EMT, undergo partial EMT. We've shown how you can do this and think about doing it by epigenetic marks. There are other things you can do. Ongoing work in Mani's lab suggests you can also bias this by changing splicing factors. There's a splicing factor called ESRP2, which changes different enzymes as part of a splicing enzyme, which affects EMT that somehow also can be used to bias this. We don't understand that at all yet, but there is some data about that. So epigenetics is not the only thing that bias you, but it's one of the important things that will. And this is more the summary, and these are some of the people who did the work. So the work on most of what just was up there is Ali Hadawi, who was a math lecturer at Northeastern, and Shubham was my student, who's now at Yale, and Sumitava is a person who is a postdoc at Rice, and then there's Yu Chen from Dana-Farber, and experimental work from Yun Zhang at Peking University now. And with that, thank you. Happy to answer any other questions. I'm done. Yes.
So we have genetic model.
The amount of which? Yes, yes. So in other words, so the time that's important is how much time it needs to spend on, for example, in the limit where the enzyme can come and immediately do its work and then leave. This doesn't work because you need to have this dilution effect. So to the extent that you need something which is not completely what people think of as enzymatic activity. Now we know because when you do these ChIP-seq experiments, you do see these activators bound to the DNA at their promoter level. So and I mean, we didn't make this up, this is just true, that these enzymes first come and bind to these regions and stay bound for some amount of time while they're doing this writing of these marks. That's a necessary part of this picture, absolutely must be put in. Yes. So the answer is, I'm sure the answer is yes, but this is the only one we've worked on. And as I said, the number of times people have tried to do this is very limited. There are papers which talk about the details of the epigenetic regulation. There are people who've tried to make models of that and try to understand multi-state transitions as a function of different enzymatic concentrations, as a function of other feedback loops. So there are pure epigenetic regulatory models. But I don't know of too many people who've tried to do this, which is to try to create a model that couples the two halves. And of course, this is back at the level of this, right? So this was our level of model in the year 2013, and now, as of where is she? Now the number of the EMT circuits have. I used to till yesterday I thought they had gone up to a size 150 nodes. Now they're up to 300 nodes. So our understanding of what are the components and what are the networks and what are the coupling has improved tremendously over that period of time. And if you would write down a paper with this circuit now, people would not even look at it. They would just laugh at you. But this is what we started with. So now we've gone back to where we started and created something at the same level of the epigenetics in the system. And now, of course, it will be necessary to look at new examples, look at extending this to other epigenetic marks, to other features that are going on, and to expand how we do this, but hopefully keeping some of the fundamental insights that we've learned. That's our perspective. So there's a lot of work to do as we look at other systems, but in all those systems, we expect the epigenetic effects to, the way we describe it is, it sort of modulates the landscape of which the cell fate decisions are taking place. The cell fates themselves are mostly determined by the genetic networks and their transcription factor interactions, but the couplings of that network can depend on epigenetics. They can be modulated by changing epigenetics. They can be inherited from one generation to the other by epigenetic inheritance, which occurs with a longer time scale perhaps than just transcription factor levels. So we think this is relevant for, basically will be relevant for many, many systems. Yeah. Is there another system like that? So the other system, which is sort of like that, the other case where there are some examples where people claim to see, not exactly that, but there are cases where this competition between different sites for what you care about and the rest of the thing is important. In microRNA, for example, you have a microRNA, and it also has a very large footprint. You can have many, many targets. And so when you, let's say, overexpress some microRNA in your system, you can not get much of an answer because most of it is going to, you're expecting that you're suppressing EMT, but in fact, most of it is going somewhere else. So people have seen effects like that, but not so much this very counterintuitive effect, right? So effects where you get no phenotype because essentially you think you're changing something locally, but really the effect is being diluted globally. People have seen that in the microRNA concept. There you're really talking about getting rid of inhibitors for the most part. So you're trying to inhibit something, but you're not succeeding. But I don't know, maybe other people know of examples like this, but we were struck, as I said, I'm struck by the fact that when I looked at this paper and listened to, was shown this movie, I said, how the hell can that happen? So that's enough to get me started, at least thinking about the problem. Yeah. Yes. Yeah. Yeah, so that's the type of experiments we have where you do this serial change. Where serial means one after the other. You do something first, and then you do the second thing afterwards. And it can depend on the order you do it, because you can have hysteresis in what the state is of these marks if you have feedback in the network. So the answer is yes. So we have there's a whole list of predictions you can make, and then the issue is convincing the experimentalists to test some of those to make sure that they're correct, or whether we need to generalize what we say. So yes, so that's the way the status is right now, is for this particular system, we think we're happy, subject to what new experiments show, and now we're trying to understand how to think about this more generally in other systems. Okay, everybody needs coffee, especially me. So, okay.